Welcome to Module 2 of our course on medical hematology, bacteriology, and parasitology. In this module, we turn our attention to the microscopic world of bacteria and parasitic organisms, two major groups of pathogens that profoundly impact human health. We will explore their structure, life cycles, disease mechanisms, and laboratory identification, as well as how to prevent and control the diseases they cause. By the end of this module, you will understand not only the scientific details but also the clinical and public health relevance of these organisms. Part A. Bacteriology. 1. Historical perspectives of bacteriology. Bacteriology is the branch of microbiology dedicated to the study of bacteria, single-celled prokaryotic organisms. But our knowledge of them is relatively recent in human history. In the 17th century, Antony van Leeuwenhoek used simple microscopes to observe animalcules in pond water, laying the foundation for microbiology. By the 19th century, scientists like Louis Pasteur and Robert Koch demonstrated that microorganisms were responsible for specific diseases, leading to the germ theory of disease. This period was known as the Golden Age of bacteriology, during which many pathogenic bacteria were discovered and linked to diseases such as tuberculosis, cholera, and anthrax. 2. Discovery of microorganisms. The discovery of microorganisms transformed medicine. The ability to visualize, culture, and identify bacteria allowed scientists to develop vaccines, antibiotics, and sterilization methods. Without this understanding, controlling infectious diseases would be nearly impossible. 3. Definition and scope of bacteriology. Bacteriology covers a wide scope. Classification of bacteria into groups based on morphology, staining, and genetics. Physiology, understanding how bacteria grow, metabolize, and reproduce. Pathogenesis, how they cause disease. Epidemiology, how bacterial diseases spread. Prevention and treatment, vaccines, antibiotics, and sanitation. 4. Morphology and fine structure of bacteria. Bacteria vary in size, shape, and arrangement. Shapes: cocci, spherical; bacilli, rod-shaped; spirilla, spiral. Arrangements: diplococci, pairs; streptococci, chains; staphylococci, clusters. Under the microscope, morphology is a starting point for identification, but it's the internal and external structures that truly define bacterial capabilities. 5. Bacterial structures and functions. External structures include flagella, for motility; pili or fimbriae, for attachment to surfaces or exchange of genetic material; capsules, polysaccharide layers that protect against immune attack. Internal structures include cytoplasm, site of metabolic activity; nucleoid, region containing bacterial DNA; ribosomes, for protein synthesis; inclusion bodies, storage of nutrients. 6. The bacterial cell wall. One of the most important features in bacteriology is the cell wall, which maintains shape and prevents rupture. Two major types exist. Gram-positive bacteria, thick peptidoglycan layer, retain crystal violet stain. Gram-negative bacteria, thin peptidoglycan layer, outer membrane with lipopolysaccharide, appear pink after gram staining. This distinction is critical because it influences antibiotic susceptibility and pathogenic mechanisms. 7. Relationship between size and metabolism of bacteria. Smaller bacteria often have a higher surface area to volume ratio, allowing faster nutrient uptake and waste removal. This can translate to faster growth rates, which is one reason bacterial infections can progress rapidly. 8. Nutritional requirements of bacteria. Bacteria need carbon, oxygen, hydrogen, and electrons for energy and growth. Macronutrients: C, O, H, S, P, N, K+, Ca superscript 2, Mg superscript 2, Fe superscript 2, Fe superscript 3. Micronutrients: Mn superscript 2, Zn, Co superscript 2, Mo superscript 2, Cu, etc., often serving as cofactors for enzymes. Some bacteria require oxygen, aerobes, while others grow without it, anaerobes. Some can switch depending on conditions, facultative anaerobes. Part B, Parasitology. 1. Introduction to parasites. Parasites are organisms that live in or on a host and derive nutrients at the host's expense. In human medicine, parasites include protozoa, helminths, worms, and ectoparasites like lice or ticks. 2. Classification of protozoa. Protozoa are single-celled eukaryotic organisms. They are classified based on locomotion. Amoebas move by pseudopodia, e.g., Entamoeba histolytica. Ciliates move by cilia, e.g., Balantidium coli. Flagellates move by flagella, e.g., Giardia lamblia. Each group contains pathogenic species responsible for diseases such as amoebiasis, balantidiasis, and giardiasis. 3. Classification of nematodes. Nematodes are roundworms. Common human pathogens include Ascaris lumbricoides, causes intestinal obstruction. Strongyloides tecoralis, can cause severe hyperinfection. Trichuris trichura, whipworm infection. Dracunculus medinensis, guinea worm. Trichinella spiralis, causes trichinosis. Enterobius vermicularis, pinworm, common in children. 4. Life cycle and pathogenicity of cestodes. Cestodes are tapeworms, flat segmented worms. Tinea solium and Tinea saginata infect humans via undercooked pork or beef. Larval stages can cause cysticercosis in the brain or muscles. The life cycle typically involves a definitive host, where the adult worm lives, an intermediate host, which harbor larval forms. 5. Life cycle and pathogenicity of trematodes. Trematodes, or flukes, are flatworms with leaf-shaped bodies. Schistosomes cause schistosomiasis, affecting the liver, intestines, or bladder. Fasciella hepatica causes liver fluke infection. Paragonimus westermani infects the lungs. These parasites often require snails as intermediate hosts. 6. Methods of demonstrating parasites in clinical samples. Laboratory detection depends on the suspected organism. Blood, thick and thin smears for malaria parasites. Feces, stool microscopy for eggs, cysts, or larvae. Urine, detection of Schistosoma hematobium eggs. Vaginal swabs, for Trichomonas vaginalis. Skin snips, for microfilariae in onchocerciasis. 7. Mechanisms of disease production. Parasites cause disease through mechanical damage, blockage of blood vessels or intestines. Nutrient depletion, consuming host resources. Toxic effects, releasing harmful metabolic products. Immune modulation, evading or suppressing the immune system. 8. Epidemiology and control of parasitic diseases. Control measures include sanitation, safe disposal of human waste to prevent fecal contamination. Vector control, reducing mosquito populations for malaria prevention. Mass drug administration, treating entire communities. Health education, promoting hygiene and safe food practices.